Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 44 of chapter 15, the discovery of auxin, part 2. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. In the previous lessons regarding to the chemicals which helps phototropism, there are some questions which are unsolved, such as what is that chemical signal? Now, let's take a look at what exactly is the chemical signal. The substance or chemical that controls the phototropic response in the plant is actually a plant hormone. In general, we call them the auxin. Auxin is a general term for the plant growth hormone. One of the examples of auxin is indole acetic acid. Indole acetic acid, or in short form we call it IAA. But I think the most important thing is remember the word auxin. It's a general term for growth hormone in the plant. Auxins, they are produced in very small amount in the apical meristem. It helps to promote the primary growth in the region of elongation. The second unsolved question is, what happens to the chemical on the bright side? Does the chemical evaporate and gone away? Are they destroyed by the light? Do they move away from the light to the shaded side? What happens to them? Let's take a look. There are two different hypotheses, 假說, that means a possible explanation. So explain why there is no auxin in the bright side. One of the hypotheses is maybe the light they destroy auxins. That's why you see no auxin on the bright side. The second hypothesis is maybe the light it causes the auxins to move away from the bright side to the shaded side. Which one is correct? Let's check. Case 1. If light destroy auxins, there are two setups, setup A and B. In both setups, we cut out the tip of the coleoptile and put it on top of an agar. If light really destroy auxin, then in setup A, under uniform light, there should be no auxin in the tip or in the agar. In the setup B, it is in the darkness. Therefore, the tip should produce the auxin and the agar should contain the auxin as well. Afterwards, the agar is put back onto the coleoptile, but only on the right-hand side. In both setup A and B, the coleoptile both bend for 24 degrees. By looking at the result, it seems that both A and setup B, they bend for the same degree. That means in the agar, it should contain the same amount of auxin. Therefore, light should not destroy auxin in this way. The first hypothesis is not correct. It's wrong. Light does not destroy auxin. Let's take a look at the second hypothesis. If the light moves the auxins away, to check whether light can move auxins away, you need two setups. In the first setup, you have setup C and D. You have the tip right on top of the agar, and in the middle, there is a mica plate, 1 sec pin, which helps to block the lateral transport of auxin. Therefore, on both left and right side of the tube, they should have the same amount of auxin after production. Therefore, in the agar on the left, the agar on the right, they should also contain the same amount of auxin. In the other setup E and F, you are going to put the tip on top of the agar. However, this time, the mica plate in the middle is not separating the left and right tube completely. Therefore, if the light really can move auxin, some of the auxin, they might move to the right-hand side. On the left-hand side, there will be less auxin. And therefore, in the agar, there should be less amount of auxin in the agar on the left, while more auxin on the right side of the agar. The agar blocks are then put on top of the coleoptile. In setup C and D, it has the same degree of bending. This is because in the agar in C and D, they have the same auxins concentration. Therefore, they show the same degree of bending. In setup E, it has a lower auxins concentration, so it has a smaller bending degree. In setup F, it has a higher auxins concentration. Therefore, it has a greater degree of bending. When you just compare the coleoptile C and D, they bend for the same degree. So therefore, they have the same amount of auxins in the block. Coleoptile F bended the most. It has the greatest bending degree for 31 degrees. So the conclusion is, auxin, it will move from the bright side to the shaded side. Let's take a look at the tube. And therefore, in the agar on the left, it will have less amount of auxin, while for the agar on the right, it will have more auxins. Based on the result of the experiment, this supported that hypothesis 2 is correct. Light can cause the auxins to move away from the bright side to the shaded side, while for hypothesis 1, it is wrong. That is the end of the video of Discovery of the Auxin Part 2. I will see you in the next lesson. Bye bye.